Good evening, Akshay. Good evening. Good evening. Good evening, Savita. Good evening, Deepika. Okay. Good evening, all. Okay. Good evening, Kavita. Okay. Okay. So, friends, as I said, 13 days we are going to do the quiz from 13 chapters. Okay, so from unit 1 to the unit 13. See, this quiz is a kind of a very good practice for you. Within this short span of time you can answer exact number of questions. See, I have designed the quiz. All of you please keep it in your mind. The questions will be ranging from 15 to 20 questions. Okay, so it depending on the time. Okay, depending on the time, we will do for 1 hour. If we can solve 15 questions, we can solve 15 questions. If we can solve 20 questions, we can solve 20 questions, right? So the most important point I want you people to read and understand is, out of 15 to 20 questions, I am requesting all of you to attempt every single question. Whether you know the answer, whether you don't know the answer, doesn't matter. Try to answer every single question. Good evening, Gautam. Good evening, Pankaj. So answer every single question. And friends, for every single question, I will give a specific period of time, whether it's 30 seconds or 1 minute, I will tell the time. I am requesting all of you spend quality amount of given time on the question, and after 30 seconds to 1 minute you have to give the answer. Okay, suppose example, if I given a answer for a particular question is 1 minute or the 60 seconds, okay, you have to answer within 60 seconds or within 1 minute. If anyone give the answer after 1 minute, those answers cannot be considered. Okay, so you forgot about the vouchers and all. You just think we have to maintain the time exactly. By that it will become a good practice for us, even in case of exam also. So if I say 60 seconds, you have to give your answer under 60 seconds. If the time goes up 61, 62, 63, I won't consider those people answers for the voucher purpose. Okay, so let's start. So every day we will try to do 15 to 20 questions, which includes part B also, which includes part C also. Okay, so let's start it. Please say, let's start it. Okay. I'll give the time. Okay. First, just confirm me everyone is able to see the question or not. See friends, after the question, I will give the crystal clear explanation of everything. Okay. First, tell me everyone is able to see the question or not. Just say yes or no, or everyone type start. Then I will go ahead. Everyone type start. Then we will go ahead. Anyway, from tomorrow onwards, I will keep the timer here itself. Today I couldn't arrange it. By that you can get to know easily. Okay, can we start? Just type start. We will go it. Okay, perfect. Your time starts now. 1 minute for the question number 1. 1 minute. You have to give me the answer within 1 minute. When I say stop, after that I won't encourage anyone answers. Try it, try it, try it. "30 seconds more." "15 seconds more. You have to give me the answers. "5 seconds more. stop. Please type your answers. Please type your answers fast. Okay. Okay. So Gautam, Tarun, as well as Sumaya. Okay. We will take only the people who has given the answer very quickly. I will take only for the first 3 people only. Keep that in your mind. So friends, all of you listen once again. I am reminding to all of you, when I say stop, immediately you have to give the answer very immediately. Then I will consider only first 3 people. Okay, here comes the answer for this particular question. So we know that the answer will be what, friends? The answer for this particular question will be a D. So let me quickly explain the question. So what is the exact question that they have given, friends? Which of the following techniques is used to locate disulphide bonds in a protein? So you know, or I mean to say, we know about this particular two-dimensional electrophoresis or 2D gel electrophoresis is the one which we used for determining the I mean to say, for locating or determining the disulphide linkages. So remember, the correct answer will be D. Wherever the explanation is required, I will be explaining the clear-cut explanation also. It's a direct question, just it's a name of the technique, you can easily answer. Good. Okay, that's about question number 1. Your question number 2 is going to start. Once again, I am reminding to all of you friends, once I say stop, immediately everyone has to give the answer. Please keep that particular point in your mind. When I say stop, you have to give the answer. Your time starts now. Your time starts now. Thirty seconds more. Seconds more. 20 seconds more. 10 seconds. 5 seconds. Stop. Please type your answers quickly. I will take only first 3 people who typed answer very fastly. Okay, good. Anyway, I'll publish the results after. Good. The correct answer will be A. Friends, why the correct answer will be A? We already know that when ATP is the one which gives the highest energy when compared to the ADP and AMP. We also know very well, friends, when we break down ATP into ADP, how much kilojoules of energy we will get? We will get 7.3 kilojoules. Same way, when we break down the ADP into AMP, we will get the same kilojoules of energy. But friends, what is the point you have to remember when compared to the diphosphate, when compared to the monophosphate? Always keep it in your mind, the ATP, which is extremely thermodynamically stable structure. So please kindly do not forget this point, that is ATP can act as a highest energy precursor. At the same time, ATP is also acting like a thermodynamically stable molecule. Do not forget the statement. Yes. Next, question number 3. I will tell the time. Your time starts now. Your time starts now. 30 seconds more. 30 seconds more. 30 seconds more. 15 seconds more. 5 seconds. Stop. Type your answers. Type your answers. Oh my God, most of the people has given the wrong answer. The answer is the biotin. Oh, most of the people has given the wrong answer. Friends, what's the question asked here? The question says that the enzyme acetyl-CoA carboxylase will involved in this particular reaction. Now the question asked is, which of the following vitamin coenzyme is most likely to participate in this particular reaction? That is the question asked. I think Gautam is the one who has given the correct answer, followed by the Deepika. Okay, but we don't consider the Deepika and Gautam because you have given the answer very lately. I want you to give answer very correctly. That's okay, no problem. Forget about it. The correct answer will be what, friends? Biotin. Why? Because biotin is one of the major cofactor that will be used by the acetyl-CoA carboxylase enzyme. Right? Hence the correct answer will be D. Next question. Question number 4. Your time starts now. It's a very logical question. "40 seconds more. 40 seconds more. 30 seconds more. 20 seconds more. 10 seconds more. 2 seconds. Stop. Type your answers. Type your answers. Okay, Tarun, I think you can see it very clearly. I was aside only. Okay, the question says that when the bacteria, I'm so sorry. The question says that when the bacteria starts growing at 20 degrees centigrade or warmed at 37 degrees centigrade, they are most likely to synthesize membrane lipids with more. First of all, I am asking a question. Everyone has to answer my question. When the temperature is low, what kind of fatty acids will be present? Savita, quickly tell me. Akshay, Deepika, what kind of Sumaya, what kind of fatty acids present when the membrane is kept at a low temperature? You all know very well, unsaturated. Yes or no? When the temperature is more, what kind of fatty acids will be present in the membrane? When the temperature is high, obviously friends, saturated fatty acids will be present. See friends, you all know very well, when the membrane temperature is when the membrane was kept in the low temperature, you all know very well, right? The unsaturated fatty acid, which is having a kink-like structure. Because of this kink structure, what happened? It increased the fluidity. But when you kept in the higher temperature, the unsaturated fatty acid do not have any active role. So which will be required for the higher temperature? We discussed in the classroom many a time. We always require saturated fatty acids. Most of the people has given the wrong answer. Hence the correct statement will be, friends, option C. For your better understanding, I have kept a crystal clear explanation also. You all can read the explanation. If you have any doubt, you can ask me. So the correct answer will be C, long chain fatty acids, saturated fatty acids. Okay. Question number 5. Your time starts now. Go. Your time starts now. That's a good important question. You can answer it carefully. For this question, I will give 1 minute 20 seconds. You can go ahead. I will tell the time. $30 seconds. 1 minute more. I'll give 1 minute 30 seconds for this question. I have explained many times this question in the this concept in the exam classroom. $30 seconds more. 30 seconds more. $20 seconds more. 10 seconds more. Please think and answer. 5 seconds more. Stop. You can type the answers. Oh my god. No one has given the correct answer. The question says that, all of you look into the question. The glycolysis and citric acid cycles are important pathways to generate energy in the cell. Given below are the statements regarding the production of ATP. We read many times this particular question. Electrons released during the oxidative step of glycolysis. Citric acid cycle produce 10 molecules of NADH and 2 molecules of FADH. Absolutely a true statement. Because friends, we have studied many times. What we studied? Glycolysis give rise to how many NADH? It give rise to 2 NADH. If you look into the citric acid cycle, 1 acetyl-CoA can give rise to 3 NADH. Then 2 acetyl-CoA give rise to 6 NADH. 6 plus 2, 8. And you all know very well, if the pyruvate wants to enter into the mitochondria to convert acetyl-CoA, there is a reaction occur. We read many times. What do we call it as? Pyruvate decarboxylation, where the pyruvate is going to be converted into acetyl-CoA. We discussed many times. When the pyruvate is converted into acetyl-CoA, once again there is a 2 molecules of NADH will be produced. We read many times, friends. So 2 NADH in the glycolysis, 2 NADH when the pyruvate is converting into the acetyl-CoA, 6 NADH molecule in the Krebs cycle, and glycolysis no FADH, pyruvate decarboxylation no FADH, only in the Krebs cycle for 1 acetyl-CoA 1 FADH2, for 2 acetyl-CoA 2 FADH2. Hence 10 molecules of NADH and 2 molecules of FADH2 is going to produce. So hence option A is absolutely true statement. Now option B, electrons released during the oxidative step of glycolysis produce 20 molecules of NADH, 4 molecules of FADH. Wrong statement. Hence option C, the coenzymes produced are oxidized by the electron transfer chain. Yeah, so the coenzymes produced are oxidized by the electron transport chain. No. Now, the conversion of ADP and PI to the conversion of ADP to PI converts into ATP. This reaction is taking place in the intermembrane space of the mitochondria. See, I can say to all of you, this option D is a kind of a correct statement only. There is no doubt about it. But since I have asked most appropriate only single statement, why this is not most appropriate, let me tell you. The reason, because conversion of ADP and PI to convert into ATP, it will take place between inner to intermembrane space. What, friends? It will occur between inner to intermembrane space. if I give the question which is a combination of statements, undoubtedly you can choose option A also, as well as friends option D also. There is no doubt. But since I did not kept the inner membrane and I have given only the intermembrane space, this statement is not more appropriate. Which is the correct one is what, friends? Obviously the answer will be A. ATP cannot produced in the matrix, dear. That's wrong. ATP always produced in the inner membrane to intermembrane space. Hence the correct statement will be A. Next, 30 seconds. Your time starts now. 30 seconds. It's a direct question. 30 seconds. Ten seconds more. 10 seconds more. 5 seconds more. Stop. Type your answer. Please type your answer. The question says that type your answers quickly, everyone. An antibiotic that resembles the 3' end of the charged tRNA molecule is. See, what was that? Puromycin. Absolutely true. The option C will be the correct. Savita, where are you keeping your brain? Huh? How does the streptomycin look like tRNA, dear? Huh? Where you forgot? Puromycin is the antibiotic, right? Which looks like a tRNA. Huh? Where you forgot all. So puromycin is an antibiotic protein synthesis inhibitor which causes the premature chain termination because, friends, it resembles the 3' end of the amino acylated tRNA. Please kindly do not forget this particular statement. Friends, this is a very logical question. Think and answer. Your time starts now. For this question, I am giving 1 minute 30 seconds. Your time starts now. Your time starts now. One minute more. One minute more. Okay, no. 45 seconds more. 45 seconds more. But Tarun, I am side only. I don't know why it is causing trouble to you only. Everyone can see it. I am absolutely side, Tarun. Oh, I'm so sorry. Yeah. 25 seconds more. I'm absolutely side, Tarun. 15 seconds more. At least I am expecting one fellow to give the correct answer. At least one. 5 seconds more. 5 seconds more. Stop. Yes, good. Deepika and Savita, Akash. 3 people has given the correct answer. Very good. Very good. So what will be the answer, friends? The answer will be D. So why ammonia, sulfur dioxide, carbon dioxide and oxygen? So let's look into the explanation very clearly. So look into this. The polarity of ammonia molecule and their ability to form hydrogen bonds, it explains to some extent the high solubility of ammonia in the water. You also know very well, right? Ammonia especially can transport to the various cells by using the aquaporins. Right? So what about the solubility of sulfur dioxide in water is highly temperature dependent, and it is about 400 times more soluble at a particular temperature. And you know, right, we read in the ecology chapter also very clearly what we use it to say, which is the major gas for the acid rain to occur. We read many a times. Sulfur dioxide is the major component of the acid rain, since it mixes with water vapor in the atmosphere reacting to produce sulfuric acid. What about the carbon dioxide? Carbon dioxide is soluble because water molecules are attracted to these polar areas. Right. Then what about oxygen? Oxygen solubility is strongly same, temperature dependent. And when the temperature is becoming high, even we read in the plant physiology C2 cycle also, when the temperature increases, what happens, friends? The solubility of oxygen basically decreases. Which is increases? Carbon dioxide. We read very clearly, at a higher temperature carbon dioxide is completely solubilizing, oxygen is not solubilizing completely. So that's why Rubisco fixed the oxygen, which leads to the photorespiration. Good evening, Rachuri. Hence the correct answer will be D. Friends, once again you can check the answers with the clear-cut explanations. You can ask me anything, whatever the doubt you have. The entire PPT can be provided in the Telegram. Don't need to worry. Okay, this is a very logical question, friends. Your time starts now. 1 minute. Tarun, I hope you can see it very clearly. I'm not at all with the board. Thirty seconds more. Good evening, Saima. Please continue. 20 seconds. Fifteen seconds. 10 seconds. 5 seconds. Stop. Type your answer. Type your answer. Okay. Tarun said B. Gautam said B. Okay. What about others? Why others did not type it? Okay. Now let's try to understand the question. They are asking a question. Question says that following are the 3 single-stranded DNA sequences that form secondary structures. So this is one sequence, this is one sequence, this is another sequence. What's the question they asked? Based on their stability, which one is correct? You all commonly know that when compared to the adenine and thymine, guanine and cytosine, these two are extremely stable, more stable in nature. Now you look into this, among these 3 options, where the GC content is more, obviously GC content is more here. Then followed by option A, then followed by the option B. Very simple. Now there is a small confusion in the A and B. The GC content is a kind of equal, but here more number of what is there, adenine and thymine is there in the B. So that's why it will be less stable when compared to this particular A. Hence the correct answer will be CAB. That is option B. Very good. So I have given the explanation also. For any kind of a doubt, you can refer the explanation and you can ask me all the time. Your time starts now. 1 minute. Your time starts now. So very simple part B question. 40 seconds more. Seconds more. Oh, super fast someone. Gautam, Saima, Akshay. 20 seconds more. 10 seconds. Stop. Okay, so the correct answer for this is B. You know, it's a very simple part B question. What the Michaelis-Menten equation basically explaining us, enzyme combined with the substrate going to give rise to what, friends? Enzyme substrate complex. So it's a direct statement which was given by the Michaelis-Menten. Okay, your time starts now. 1 minute, your time starts now. This is 1. This is 1. It's a 1 by V. 1 by V. 1 by V. I'll just write it. This is 1 by V. "40 seconds more." Shrikant, Akshay, think and answer, 20 seconds more. I am asking the reciprocal. 10 seconds more. So very simple. 5 seconds more. Stop. Please type your answers. 10 is C. Oh my God, how C? See friends, what? All of you look into this. Listen, the Michaelis-Menten equation can be transformed into a linear equation by taking what? Reciprocal of both sides. You know, right? We read in the classroom many a times. What is the reciprocal equation for the Michaelis-Menten? That is only we can call Lineweaver-Burke plot. Correct na? We will just do the opposite. What are the reciprocal? Then obviously it will be 1 by V. It's a direct question. What's difficulty in that? I'm not getting you. Okay. So what will be the answer here? Obviously the answer will be option A. Correct, Gautam. Correct. Savita given correct answer. Akash, Sumaya, good. SSV, good. Simple question, don't make it wrong. Yes. Question number 11. Your time starts now. 1 minute. Question number 11. An allosteric binding site. Direct question, friends. 45 seconds more. I'm giving much time also. See, my all intention is that you have to maintain the time. That is all my intention. The vouchers or everything is separate. 30 seconds more. "20 seconds more. You have to think it. 10 seconds more. It's an easy question. Oh my God. Stop. I'm expecting at least one fellow to give the answer. Oh, very good, Akash. Very good. Fantastic. So the answer will be A. Friends, why others are not giving the answers? It's a right or wrong. You people try to give the answers. Do not worry. An allosteric binding site. What is allosteric binding site? Everyone tell me. This is an enzyme. Correct na? Here who will come and attaches? Substrate will come and attaches. This is what site we call it as? Active site. Correct na? Now, if any inhibitor or any drug, if it is acting on a different site other than the active site. Suppose a drug comes and attaches here. A drug comes and attaches here, but it is blocking the active site. How it will block the active site? By just changing the shape. Very simple. Allosteric site, in one line I will tell you. Allosteric site is the other than the active site. Even though a drug or inhibitor are associated with the allosteric site, still it can easily block the active site of enzyme. How? By changing its shape. Suppose all of you quickly recall, this is the enzyme. Here the substrate will come and attaches. But where this particular allosteric inhibitor will attach somewhere here. But now see here, the enzyme became like this. When the enzyme became like this, how does the substrate can come and attach us? Never. So it is a binding site which is separated from the active site. It is separated from the active site and affects the activity of an enzyme when it is occupied by a ligand. Very, very, very simple question. Yes. Good, 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 good. Question number 12. Your time starts now. 1 minute. It's a direct question, very simple question. You can answer just like that. My friends, look into the question. They are asking except. "30 seconds more." Okay, I'll give 5 seconds extra for this question. 25 seconds more. Question number 12, answer is B. Okay, who has given the correct answer very fast? Tarun. 10 seconds more. Gautam. Stop. Please give your answers. Please give your answers. Okay. Very good, friends. It's a very, 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 very direct question. All of the following statement in relation to the Ramachandran plot are except, because see here, they have given almost all the peptide bonds in the protein are in the cis conformation, which is absolutely a wrong statement. Very, very direct question it is. Next. Question number 13. Okay, 1 minute. Your time starts now. It's a direct question before 1. Question number 13. Your time starts now. "30 seconds more. Think and answer. Think and answer. "20 seconds more. Someone has already given the answer. Who has given? Kavita Akshaya. Okay. 10 seconds more. Question number 13. Question number 13. 5 seconds more. Stop. Stop. Please type your answers. Please type your answers. Why do bisphosphoglycerate cannot bind to oxygenated R state of hemoglobin? You know very well, hemoglobin can exhibit it in a 2 states. One we called R state and another one we called as a T state. Always remember, in the R state, binding pocket where the 2,3 bisphosphoglycerate will come and associate, it will be small. In the T state, it will be a large. So that's why, friends, 2,3 bisphosphoglycerate associated with the hemoglobin only in the T conformation or only in the T state. It cannot associated with the hemoglobin when it is in the R state. Hence the correct answer will be B. Its binding pocket becomes too small to accommodate BPZ. Very simple question. Slowly think, analyze and give me the answers. Your time starts now. 30 seconds. 30 seconds. Your time starts now. 15 seconds more. 15 seconds more. Ten seconds more. It's a direct question, friends. 5 seconds. Stop. Please give the answer. Anyone? What about people? The protein is likely to be the strongest hydrogen bond donor. Anyone? Question number 14. Obviously, it is the ammonium ion. Very good. Because friends, all of you look into this. The best hydrogen bond donor will be the one where the hydrogen is the most electrons deficient. So what you can observe in the ammonium ion, the nitrogen has a positive charge, and this in turn will make the attached hydrogen electron deficient. Hence obviously the answer will be ammonia. Good, perfect, perfect. Yes. Question number 15. Your time starts now. 1 minute. Question number 15. Thirty seconds more. Twenty seconds. You have to think and answer. 10 seconds. Stop. Type your answers. Type your answers. Question number Tarun said 15 A, Sumaya A, Gautam A. Okay. Look into the question. Why in the case of anaerobic cells, the ratio of pyruvate or lactate is much less than 1, although under aerobic condition, ratio of pyruvate or lactate is much greater than 1. What is the reason behind that? You all know very well, our body starts converting the pyruvate into lactate only in the absence of oxygen, or only under anaerobic conditions only. That's why friends, pyruvate can be converted into lactate only under anaerobic conditions. That's why friends, it is always less than 1. Very good. Okay, but Tarun, others can see. What's the problem? Okay, Pankaj. Sorry, okay, Tarun, I will do it. Okay. The answer for the question number 15 is A. I'm sorry, I'll go out completely. Then only I'll come. Okay, question number 16. Oh, sorry. Question number 16. Your time starts now. Question number 16. It's a very logical question, friends. You have to choose it. I hope, Tarun, you can see it very clearly now. Oh, others are not responding, that's all. Okay. Tarun, I hope now you can see it very clearly, right? I feel. Please let me know if you find any problem. 30 seconds more. It's a good question. You have to think and answer. They are asking a correct definition. No, think and answer. Think and answer. 10 seconds more. Five seconds more. Stop. Okay, tell me the answers, anyone. At least try to guess it. You're welcome, Tarun. You're welcome. So the question is, which of the following statements correctly defines the Salvage pathway for the synthesis of purine nucleotide. We read many a times purines or pyrimidines, they can be synthesized by two pathways. One we called as Salvage pathway, one we called as de novo synthesis pathway. See friends, in case of de novo synthesis pathway, we have to spend a great amount of energy. But when you look into the Salvage pathway, here also we will spend the energy, there is no doubt about it. But when compared to the de novo synthesis, the amount of energy that we spend is extremely low. So that's why we can say the Salvage pathway for the synthesis of purine nucleotides is energetically advantageous. Yes, it doesn't mean that we are not spending energy. It means that we are spending energy, but compared to the de novo synthesis, it is less. That's the answer. Okay, that's the point. Hence, question number 16, option is C. You can read the explanation and you can ask me for further doubts. Okay. Your time starts now. Question number 17. Every day I'll make very tough questions. Since it's a first day, very simple questions. Anyway, it depends on your preparation. 30 seconds more. It's a logical question, try it. It's a very good question, friends. 10 seconds more. Okay, someone has given correct answer. Great. Savita, Sumaya. 5 seconds more. Stop. Please give answers. Please give answers. Tarun, we can't say C or D, no. It's only one option you have to give. Good SSV. So what is the role of internal ribosome entry? It is the one actually promoting the translation under the adverse conditions. So the correct answer will be, friends, option C. So an internal ribosome entry site, abbreviated IRES, is a nucleotide sequence that allow for translation initiation in the middle of mRNA sequence. As a part of the greater process of protein synthesis, usually in eukaryotes, translation can be initiated only at the 5' end of the mRNA molecule, since 5' cap recognition is required for the assembly of initiation complex. It only occurs during the adverse conditions only. Let me know, I am going explanation little quickly because I kept it very clearly. If you have doubt, you can ask me immediately. Okay, 30 seconds. Question number 18. 30 seconds. 30 seconds. Question number 18. How? 5 seconds more. 5 seconds more. Stop. Stop, 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 stop. See, it's a very simple question, right? Why does the DNA twist? Obviously because of the three-dimensional structure. We all know that the three-dimensional structure is the most complex structure. In the complex structures only the twisting occurs. Even you look into the protein, where the protein folding occur. Protein folding also occur majorly in the complicated folding, I mean to say. Protein folding also occur in the three-dimensional only. Then obviously answer will be B. Yes, 30 seconds. Question number 19, 30 seconds. Dash molecule is involved in the regulation of gene expression. 30 seconds. Gene expression, whether it is a gene silencing also, whether it is gene promotion also, Gene stimulation also doesn't matter. Which is involved in the regulation? 10 seconds more. 10 seconds more. Okay, stop your answer. Okay. Dash molecule is involved in the regulation of gene expression. There is a no doubt. Most of the students got a doubt between the A and B. Okay. See, RNA is the one where the gene expression starts, but RNA cannot do the gene expression on its own. It requires compulsory tRNA, which brings the amino acid, which requires the rRNA, which brings the ribosomes and involved in the synthesis of protein. miRNA, this is the one, my friends, majorly involved in the gene silencing mechanism. We can also call it as RNA interference technology. Correct, na? Is the correct answer will be B. Okay, last question. Okay. 1 minute. Here is the question. Question number 20. 1 minute. Question number 20. Question number 20. 40 seconds more. Shreya, whatever you said is absolutely true, dear. Yes, that's what you can say. Correct. RNA for the gene expression, and you can say that mRNA is for the gene regulation. Whenever, dear, you have to listen, whatever you said is right. The gene, I'm so sorry. I'm extremely sorry. Yeah, please go with the answer. Okay, stop. Yeah, the answer will be X-ray diffraction measurement. And Shreya, when it comes to your question, there is a no doubt mRNA is the, or RNA is the one, or RNA transcription is the site where the gene expression will start. But when you look into the mRNA, it will regulate whether the gene has to be expressed at the higher level, or gene has to be regulated, or gene has to be silenced. That can be decided by the mRNA. So friends, when it comes to this particular question, what will be the answer? Answer C. Absolutely true. Right? It's a very, very direct question. Okay. Because X-ray crystallography is the structure which is used for identifying the atomic and molecular structure of a crystal. It's a very direct question. Okay. So that's all for the 20 questions. Okay. I hope the discussion will help you a little bit. My only aim is that you have to maintain the specific time for the question. By that it will become a good habit in the examination hall which we are going to face after 15 days. Okay. So friends, please prepare for the tomorrow. Tomorrow we are going to do the cell biology. I will make in the cell biology 10 easy questions, and I will make 10 difficult questions and time consuming questions. What is our goal? Whatever the questions that are time consuming, you have to do it very, very faster. Okay. So we will announce the today's winner in the by tomorrow in the Telegram group. Okay. So once we announce the winner, I will share the 500 rupees Amazon voucher to that particular person, either through their respective email ID. Okay. So Amazon vouchers, these are all kind of encouraging things. My intention of conducting quiz is time management. If you keep on managing a specific minute for the one question, that will help you a lot in the upcoming exam. Okay. I hope the discussion is little useful for you, all of you. Yes. Okay. I will see you in the tomorrow class sharp 7:30 PM with the cell biology. Thank you all. Please kindly share to most of your friends by that they can also utilize this. Thank you all. Thank you.